So we'll go ahead now and move to my colleague, Erin Gray, who will introduce the next session.
Hello, good afternoon, everyone. So after that great discussion, we are going to switch gears a bit and move to our last keynote presentation, followed by the final panel discussion. So maybe you need to just kind of stretch out a little bit and refocus. So I'm Erin Gray. I'm a program director at the NIA in the Division of Neuroscience, and I'm very pleased to introduce our next keynote speaker, Dr. Karel Svoboda. Dr. Svoboda is the director of the Neural Dynamics Unit at the Allen Institute, where a current focus of his research is to identify core principles and mechanisms underlying information processing in brain-wide neural circuits. Dr. Svoboda also has a longstanding interest in new molecular, optical, and computational methods to map neural circuits and interrogate neural function in intact brains. For today's keynote, Dr. Svoboda will discuss cell type specific analysis of neural circuits and their dynamics at the Allen Institute for Neural Dynamics. We should have time for Q&A following the presentation, so please do remember that you can raise your hand during the Q&A or enter questions into the chat. So please go ahead, Dr. Svoboda.
Good.
We can hear you.
Great. Well, thank you, Erin, for the introduction, and thanks to the brain staff for putting this very interesting symposium together. So today, rather than presenting a polished talk, I'll try to give you some examples of how we use brain cell atlases, that many of you have contributed to understand neural circuits and neural coding within these circuits. And throughout, I'll try to seed some discussion, open, point out kind of open areas for investment and seed a discussion of the kinds of questions that the panel already listed. Now, great advances, of course, have been made in enumerating and characterizing the different cell types that compose the mammalian brain. This is a study from about almost 10 years ago from the Allen Institute, from the anterolateral motor cortex here. And this work is mainly based on high throughput methods, right? Sequencing and multiomics. We've heard a lot about that over the last couple of days. And of course, these cell types should really be referred to as transcriptomic types right now, because we don't know yet how these transcriptomic types or T types really correspond to cell types that ultimately will be defined based on a multimodal approach, including connectivity, biophysics, and other kind of data. But how do we use this data to understand the brain at a kind of multimodal level? The T types, of course, are precisely organized in space, and you've heard a lot already and a lot of strong arguments about why spatial methods are important, right? Because T types are precisely organized in space, and they can be organized in small nuclei, like perhaps the norepinephrine neurons and locus coeruleus, just a few thousand neurons, 1,000 neurons in the mouse, maybe a couple of tens of thousands of neurons in humans in a tiny structure. Or they can be more widely distributed along cortical areas. But even here, right, the T types in the cortex, as shown here in the example here from the mouse, still limited to cortical layers. The important point here is that location in the brain tells you a lot about the T types, the cell types in the neighborhood, and the spatial variation of gene expression is on the order of 100 micrometers, or can even be less. And that's similar in larger brains, where, of course, the cortical mantle is much larger, but the layers are really not much thicker across a mammalian species, maybe a factor of 2 or 3, not orders of magnitude. Now, all other measurements, we think circuit mapping and measurements of electric activity have to index to these spatial coordinates and ultimately identify the T type corresponding to the measured neurons in different modalities. Now, the T types are we think of them as a parts list. They're necessary but not sufficient to understand the brain. In particular, cell types connect into brain-wide neural circuits, and it's these circuits really that process information and produce behavior. How does that work really on a cell type specific level? We still have only a very rudimentary understanding of the function of any specific cell type in the mammalian brain, especially in the center of the brain. Now, but importantly, I want to point out that neural circuits, and you will discuss more about this perhaps in the context of connectomics, neural circuits are organized on even a finer scale than variation in gene expression. This is just an example of many from a mesoscale analysis of circuits that just highlights that different cortico-thalamocortical circuits are interdigitated on fine spatial scales. Here, connecting motor cortex that controls orofacial movements, motor cortex that controls limb movements, with thalamic nuclei, parts of thalamic nuclei that are comprised of individual transcriptomic types. So here, kind of organization of circuits that are even on a finer scale than transcriptomic variation. So this really brings us to the first takeaways I want to mention. Common coordinate frameworks are really foundational for multimodal analysis, transcriptomics, circuits, neurophysiology. And we have to be very careful and rigorous about registration on spatial scales on the order of 100 micrometers across individual animals and also across modalities to link all these different data modalities together. And I don't think we're doing quite as good a job as we should be in our experience. Really, that registration still remains to be done. And often in some databases, the data required, the context that allows registration is not available. So we should, going forward, think very carefully about how to spatially register different spatial information across individuals and animals, since will require standardized data pipelines for registering images and image volumes to common coordinates like the CCF, and also ground truth measurements to estimate the experimental errors and perhaps even biological noise, right? Even in isogenic animals, who knows if development proceeds in exactly the same way, and how is this across maybe outbred strains and so on and so forth. And we need to develop quality metrics that allows to report how good the registration is for a particular brain workflow and modality. Finally, with respect to atlases, I think it's pretty clear that multiple parcellations and anatomical ontologies should coexist and, of course, evolve as we learn more. These are going to be based on cytoarchitecture, classical atlases, transcriptomics, lots of information there, but connectivity, and these may not always overlap in every place, as I've already shown you. All right, let's now turn to neural circuits. We're interested in particular in our institute in the neurons that connect brain regions, projection neurons that send axons out from one brain region to connect to another brain regions. And the questions that we're asking is, what are the T types that connect brain regions, the transcriptomic types, and what are their shapes? How do they connect with these different brain regions? It's just an example of why this is incredibly fruitful, especially at the level of single neuron. Okay, so here we did some single cell reconstructions in the motor cortex and discovered a couple of neurons that project out of the motor cortex, so-called pyramidal tract neurons, so brainstem projecting neurons of two types that were missed based on bulk analysis. One neuron here that projects to the thalamus, not to the medulla, where orofacial movements are initiated. Another set of neurons here that project to the medulla, but not to the thalamus. And we found later, using kind of direct methods, that these neurons are transcriptomically distinct, and using electrophysiological recordings, that these neurons do different things in controlling movement and planning movement. And so this just kind of highlights how powerful single cell reconstructions are if we can put them into the molecular context, ideally at the level of individual neurons. So how do we link single cell projections with transcriptomics? We've been setting up an imaging pipeline to do single neuron reconstructions efficiently. I'm going to go through this very quickly. This was recently published. We use something called expansion microscopy, which makes that can be done at the level of the whole brain, which makes the brain bigger for better resolution, and also makes the brain exquisitely clear for best of class imaging. Now we have a huge sample to deal with that we have to image at diffraction limited submicrometer level resolutions. We need a microscope that can image these huge tissue volumes in a rapid manner. So we've developed, and when I say here, this is really work spearheaded by Alan Glaser and Jam Chandrasekhar. In general, I've put some names in the bottom of the slides, go through many studies. I won't be able to mention all individuals throughout the talk, so please keep your eyes peeled for the names in the bottom of the slide. So we had to develop a microscope that can image these large tissue volumes without sectioning the tissue volumes. It has very large working distance on the order of 3.5 centimeters, much larger than ordinary biological microscopes, and can image much faster while retaining high contrast and high resolution. This is a modification of a light sheet microscope, makes use of a tech stack that was originally developed for inspecting semiconductor devices or flat panel TVs. And one gets exquisite images using this microscope. This is sparsely labeled sample, and you can see these incredible high contrast images, even 1.5 centimeters deep in the tissue, which then allows us to use semi-automated methods to reconstruct the axons in these sparsely labeled brains. So how do we then link these? I'll give you 2 examples of how we link these reconstructions to molecular, to transcriptomic data. I'll focus first very briefly on the norepinephrine system that I've already mentioned. Norepinephrine neurons in locus coeruleus. They're known to have participate many different functions in learning, in arousal, sleep, stress, and they are implicated in many different disorders. The major targets, of course, of neuropsychiatric medications, and they degenerate early in neurodegenerative disorders. So early on, we with our single cell reconstructions, we noticed that these neurons are extremely complex and also diverse. And so the picture that norepinephrine neurons are relatively homogeneous are already indications that it's diverse, but it's pretty striking at the level of individual neurons. Here, a couple of neurons that project to the front of the brain, to cortex and cortical nuclei. A couple other neurons that project to the cerebellum, which is, of course, involved in movement control. Another couple of neurons that project to the medulla here from the pons, where these locus coeruleus neurons are, to the medulla and to the spinal cord. And you can see here that with this co-innervation map, there are probably like 3 or 4 modes of projections in these neurons when analyzed at the level of individual neurons. Neurons that project to the front, to the back, to the spinal cord and the medulla, and somewhere in the middle of the brain here, thalamus, hypothalamus, and to the cerebellum. And these neurons are arranged in an orderly fashion in a dorsal to ventral gradient, where the dorsal neuron project to the front of the brain and the ventral neurons to the back of the brain. We can now see how these soma positions compare to gene expression profiles. And in the locus coeruleus, we find gene expression that varies essentially gradually here in PC space and also in brain space. Here in this dorsal ventral gradient, and when we kind of superpose the projection types onto this spatial gradient, we find that the dorsal population projects mainly to the front of the brain, the ventral population to the back of the brain, and the middle population somewhere in between. And I'm simplifying a very complex and comprehensive study here. So this is one way of linking kind of a graded projection map along the locus coeruleus, which really consists of one transcriptomic type in the ABC atlas, but still contains gene expression gradients to gene expression gradients. So how do we link projections of individual neurons now to, you've heard already about neurons in the medulla and the pons. They're transcriptomically very complex, interdigitated neurons belong to different transcriptomic clusters. So how do we link the individual neuron, the single neuron reconstruction to transcriptomic types in that case? So here are the projection types from the medulla to different parts of the brain. Medulla is down here. This is where the somata are localized. And I'll illustrate how we do this by focusing on the cerebellar projection neurons in the medulla, shown here in red. These are diverse. They come in many different nuclei. So here is a neuron from the external cuneate nucleus, which encodes proprioceptive information. Vestibular nucleus right here about head, cares about head movement. Here are some integrative neurons in integrative nuclei, for example, intermediate reticular nucleus of the medulla, and their projections to the cerebellum. So different neurons are located in different parts of the medulla and project to different parts of the cerebellum in stereotype with sort of stereotype projection patterns. So what we can do to link these data sets, these structural single cell reconstructions and transcriptomics, is use an intervening, a linking data set where we label all cerebellum projecting medulla neurons, perform transcriptomics of these neurons, and by transfer labeling, by label transfer rather, localize the transcriptomic clusters within the medulla. And again, localization is critical here, right? We need to localize these transcriptomic clusters here at the subclass level using the Allen Institute terminology to parts of the medulla, and then through position, link the reconstruction, right? The position of the soma of the reconstructed neuron and the location of the subclass, and allows us to assign a transcriptomic subclass to individual reconstructed neurons. And this works great in the medulla. Here, the inferior olive complex that sends learning signals to the cerebellum is linked to this transcriptomic cluster. Another that here vestibular neurons and so on and so forth, and they're the proprioceptive neurons in the cerebellum. So what are the takeaways? So linking transcriptomics and morphology connectivity can be achieved in different ways depending on the cell population. And registration to common coordinates is really foundational for this multimodal analysis. And I've kind of highlighted this for two different kinds of methods, right? Where we're mapping different projection types to gradients in gene expression, and also in the case where different T-types are intermingled in the medulla. And I think going forward, we need models of cell types that explicitly treat discrete cell types and spatial variation in gene expression and projections and other features. And we also now need cell type taxonomies and databases that incorporate multimodal data, transcriptomics and epigenomics with morphology, connectivity, and biophysics to get a more coherent, holistic view of cell types and circuits. So let me now, in the last few minutes, give you our vision of how we link neural coding and signaling to transcriptomic types. This is very much at early stages. Of course, the brain generates behavior with electrical signals that course through neural circuits. It's really about understanding how neural signals produce behavior and cognition in the context of neural circuits. And major challenge is really to link recordings of spike trains to specific transcriptomic types at scale. So one advance is we've developed a recording probe recently. This is still work in progress to combine best-of-class electrophysiology with dual channel optogenetics. So these little sites here can be used to illuminate the tissue surrounding the electrodes in a random access manner. And then if neurons are sensitive to light using channel rhabsin, and of course, we need transcriptomic and multiomic data to transduce neurons with channel rhabsin, to develop the methods to transduce neurons with channel rhabsin. So this is how this relies on the gene expression multiome atlases. So if we have transduced neurons, for example, here in the striatum, right, D1 neuron that projects maybe to the SNR intermingled with the D2 neuron that projects to the GPE, we can now tell what these neurons are on the electrodes because they're responsive to lights. And this works great with these electrodes. These electrodes really make these measurements routine, just illustrated here with a couple of D2 units, a couple of D1 units here. We can identify these units by their rapid and phasic responses to photostimulation. And typically experiments in the striatum, we can actually identify more than half of the units in recordings in many experiments, which makes sense because D1 and D2 neurons make up the majority of the cell types. So we can now put this photo tagging idea together with the analysis of structure and pin different functions in the context of learning to different projection types, for example, in the locus coeruleus that I've talked about earlier. I'm going to have to make this very quickly here. And it turns out that the dorsal neurons that project from the locus coeruleus to the cortex and other parts of the front of the brain show learning signals that resemble what we refer to as reward prediction error. The neurons are more active when the situation in a learning experiment is better than expected. And the neurons that project to the back of the brain, it's the converse. They're excited by lack of rewards. Very different learning signals that are sent by locus coeruleus neurons to different parts of the brain. Our vision going forward is now, last minute or so, we're missing some of the units, of course, that we're recording. We can only photo tag a couple of neurons at the same time. In many brain regions, there's maybe a dozen neurons in any kind of location and maybe dozens of neurons in a particular brain compartment. And so what we'd like to do is to use the optotagging data to develop ground truth data, to develop classifiers so we can recognize the neurons based on their spiking patterns and their spike shape in the absence of the optogenetic tagging. So we're producing this kind of ground truth data just illustrated here for the striatum, and this is starting to work in, for example, for cortical GABAergic interneurons, and it generalizes across individuals, and in some cases also across species. So the same electrophysiological signatures are not just found in rodents, but primates and likely also humans. And a very nice paper doing similar things in the cerebellum just came out. So our vision going forward for identifying neurons, transcriptomic cell types in the brain is to generate a lot more ground truth data using optotagging. The transcriptomic multiome data will allow us to develop tools to access specific cell types through enhancer reagents, perhaps, or transgenesis in mice. Enhancer reagents can also be used in primates. Do a lot of the optotagging in mice, then see how it generalizes in primates and ultimately in humans, and ultimately develop a cell type lookup table that will allow us to do cell type specific recording with high throughput and high multiplexing. So just gets me to my third take home message: new recording probes and reagents, enhancer AAVs and transgenic mice make identification of T types in electrophysiology routine through optotagging. This is still a low throughput method, 2 or so cell types at a time. It's also limited to model systems because it requires introducing channel rhabsin and related reagents to neurons. And there is really a great need to develop efficient, highly multiplexed and general methods for identifying T types in electrophysiology. This remains a grand challenge for brain research. We think that a cell type lookup table for electrophysiology is a promising direction. but it really requires now a coordinated effort to acquire ground truth data across the brain on a much larger scale. Okay, and let me just mention, this is the work of a team at the Institute for Neurodynamics, an incredible team of engineers, scientists, program managers that I've had the pleasure to work with. And I'd be happy to take questions if there are any. Thank you.
Great, thank you very much for that excellent talk. We are a bit tight on time, but we can take one quick question. It looks like Batsala.
I just had a question, because are you in an effort to correlate the transcriptome analysis to the cell type, to the the spatial organization of the brain? I was wondering, when you do transcriptome analysis, do you only take the transcriptome of the cell body, or do you actually take it along with all the axon data? Because the axon is actually quite extensive, like it's very long, and there's actually going to be RNA like perturbations along the axon. So I mean, are you able to get the different RNA sequences of along the axon also?
That's a good question. So let me just say that the spatial methods usually look at the transcript close in the soma, right, in the ER, or associated with the ER. And there's also relatively little transcript in the dendrites and axons. It's certainly there in dendrites, but it's sort of numerically a relatively small number. But it's possible that there's kind of an enrichment of transcripts in neurites that are not looked at in these spatial genomic methods.
Yeah, because
Thank you.
fast, like axons that need to transmit information fast, if you have like a long axon, you usually can have message close to the signal, so that you can translate ribosomes, or like can translate mRNA close to the signal, so it doesn't have to transport it from the soma to the axon.
Thank you very much. We are out of time for questions. I'm very sorry to cut it off, but please do enter your questions, comments in the chat, and Dr. Svoboda can continue the conversation there. Thank you again, Dr. Svoboda. That was an excellent talk.